Physical digestion is the physical breakdown of large food particles into smaller particles. So large food particles are broken down into smaller particles. It's just like crushing the food from big particles into smaller particles using energy. So the reason why this is happening, so that you increase the surface area, so that the chemicals or the enzymes can act upon this food very fast. And then you're saying that occurs during mastication, churning. You find these ones in the stomach. It's peristalsis, during peristalsis. When we talk about peristalsis, when the food is here is going down, we call it peristalsis. So in that period, you cannot bring the food back. If the food has gone down, you cannot tell the food that go back. No. It is just a rhythmic contraction. It's automatic contraction and relaxation of the circular muscles. So the muscles, they contract as they push food downwards. This is called peristalsis. So peristalsis is a reflex action. It means that you don't have control over it. It's triggered by presence of food in the alimentary canal, and then this is very important in transporting food and water in the body. So as food is going, you can find it in the esophagus, you can find it even in the stomach. We find them there so that food can move in one direction. This is an example of peristalsis. Here, when the food is here, these muscles contract and then this one relax. When these ones contract, they push this food downwards. So these muscles cannot relax and this one will contract. That's why even if you're upside down, still the food will move in the one direction. It will move from the mouth and then goes to the stomach. So then now it moves, then the food moves downwards. So this kind of food in form of a ball, we call it a bolus. So now the food moves, it continues to move as these muscles relax. When they relax, these ones contract, they work antagonistically. In science, we call it antagonistic muscles. When these ones contract, this one relaxes, and then the food is pushed downwards, and then the food continues in that direction. And when we talk about chemical digestion, it's a breaking down of large food compounds into smaller food compounds using digestive enzymes. So here, large food particles are broken into smaller food particles by the help of the digestive enzymes. So the enzymes, which you call the chemicals, that's why you say the chemical digestion. The enzymes, they help in breaking down this large food into smaller food. When you talk about large food, it may not be necessarily be large, but is large when compared to those particles or nutrients which are absorbed into the bloodstream. Because when you eat, when you chew, you break down from big particles into smaller particles. So now the smaller particles, those particles which you have broken down into, are what you call now the large. They become large when it comes to the chemical digestion. And then those particles are broken down into smaller particles, which can be absorbed now by into the bloodstream. And what do they do? Yes. We say that you have different groups of enzymes. We have which kind of food do they act upon, and then the end products of the food. And then lastly, which kind of pH or medium. We are saying that we have those ones found in the saliva, that's the mouth. Pancreatic, those ones are found in the duodenum. And then intestinal, those ones are found in small intestine, the ileum, the one which is coiled. If you remember the structure of digestive system. So this only act on, it means that carbohydrates are being digested in the mouth, no stomach, no. And then duodenum, and then the small intestine. So which substrate? We call it a substrate. We don't call it a food. No, we call it a substrate. You have what you call carbohydrate, which is starch. Sometimes we call it starch, but carbohydrates can also be in different forms. Then which pH is likely to be where these enzymes are working is supposed to be slightly alkaline. The reason why these ones cannot work in the stomach is because the stomach, the pH is acidic. And then what is the end product when you digest all the carbohydrates? You find out that glucose is the end product of that food. Then you have the second category of proteins, which we call the proteases. We find protein digestion in the stomach. the pancreatic juice, that is the duodenum, and then you find it in the intestinal juices. This is the small intestine, which is ileum. So which kind of substrate is being acted upon, which is food? We don't call it food, I told you. Which substrate is proteins? Which kind of pH do they work in? They work in acidic medium, that is in the stomach, and then they work in alkaline medium, that is small intestine. That's why these proteins can work in acidic and alkaline. That's why you can find them in the stomach and the small intestines as these ones. They also work in the alkaline medium, but they don't work in acidic medium. That's why you cannot find these in the stomach. You only find them in the small intestines. Then which end product, when you digest proteins, which end product is supposed to give you? Is supposed to give you what you call the amino acids or amino acids. The last group is lipase. The lipase, these are lipids. Lipases are enzymes which act upon lipids. So carbohydrates, the proteases, and then the lipases. The word ASE means an enzyme, as we will see. You see, carbohydrates, proteases, lipases. We will see. We will explain that as you continue with the science. Okay, we are saying that these one lipases, we find them in the pancreatic juice and intestinal juices. Pancreatic juice, now here we are talking about the duodenum, and then intestinal juices, we are talking about the small intestine, that is the ileum, the coiled one. So which substrate which is being acted upon? The substrate is lipids, which you call the fats and oils. So it depends on whether it is coming from the plants or it's coming from the animals. So the pH, because the duodenum and the small intestines, that is ileum, is alkaline, so they are supposed to also work in alkaline. What is the end product of the lipids? What's the end product of the fats? You get what you call glycerol and fatty acids. You see this one, or you did see this one in grade 10. Yeah. Note, enzymes are very sensitive to change in the temperature. So they can be denatured if the temperature is too high. Then they are also sensitive to pH. That's why that these enzymes, they have a specific pH in which they are supposed to work in, and then hence only work in optimal temperature and pH ranges. That it works maybe from 3 to 5, that is acidic, maybe from 8 to 10, that is alkaline, or 7, that's neutral. Or so they have a specific kind of pH they are supposed to work upon. They don't work in each and every pH. So what we have been trying to explain, this is a diagrammatic representation of what we have tried to explain here. So we are saying that you see, protein molecules is made up of many small amino acids. Remember, we said that when you break down the proteins, you produce what you call the amino acids. So when you break down using the enzyme proteases, we have different proteases enzymes. We have pepsin, trypsin, those are proteases, yes? The peptidases, all those are proteins. So they break down, then you get what you call the amino acids. Then you have the starch molecules, is made up of many glucose molecules. This one is made up of many glucose molecules. When you break down the starch molecule, you produce what you call glucose. And then which kind of enzymes are the carbohydrates? Examples of carbohydrates, you have a lot. You have maltase, you have the sucrase, and then the end product is glucose. Then you have the fat molecules, is made up of fatty acids and glycerol. This one, it looks like E. If you look at it, it looks like E. I call it E. Fatty acids, and then this yellow part is the glycerol. So the enzyme is lipase. When you break it down, you produce what you call the fatty acids, and then you produce what you call the glycerol. So basically that's it. And then let's look at now, after you have broken down this food into smaller particles, then we go to another step of the process of digestion as absorption. So now we have got the final product of these molecules. So what are the process? What is the process of absorption? How does it take place? So number one, it takes place mostly in the small intestine. We saw that the small intestines, with small intestines, you find out that the food is basically, you find the final digestion of food in small intestine. That is the ileum, leave alone the duodenum, because duodenum is also an example of small intestine. But the ileum and jejunum and ileum, you find them that the final product is found there. And then absorption also takes place there. So small intestine is suitable for absorption due to the characteristics below. We can ask you, how is the small intestine suitable for carrying out absorption? It's long, approximately 6 meters long. So that's why it is coiled. If you look at the small intestine, it's very long. It is coiled. Yes, yes. The small intestine is coiled. Is coiled like this. It's coiled. It's coiled. So when it's coiled, I think now you can see. So that's why it is long, so that it can increase the surface area for absorption. So number 2, the walls contain transverse folds. Transverse folds. So we're gonna see it when we look at the ileum. The inner wall has millions of finger-like projections called villi. So the villi, they protrude out. You're gonna look at the structure of a villus. If it's one, it's called a villus. If there are many, we call them villi. We don't call them villuses. We call them villi. So it has millions of these finger-like projections, so that it can increase the absorption of this food. Each villus contains microvilli to further increase the surface area. Is the surface area for absorption. So even the small ones, they have small other micro projections. So it means that the surface area, by the time the food passes all those folds, is supposed to be at least the important nutrients are supposed to be absorbed back to the body. Then here is the structure of a villus. So they are like this, but there are millions and millions. So structure of a villus, number 1, it has what you call the capillaries. It has what you call the racture. It has what you call the arterioles. It has what you call the venules. Okay, the venules are the small veins. Yes, when the vein break down or branches, then it becomes small, or it becomes small, it becomes venules. And then when the artery break down like small branches of artery, then we call them arterioles. So then the vein, it combines back, and then it joins the big vein, which you call the vena cava. And then these on the arteries, as they bring blood with low level of nutrients, it comes like this. Yes, you see, and then goes up. Yes, you see this color is different from this color, and then picks the nutrients. Now the nutrients starts to come in. They start to come into the blood, and then the veins goes like this. You see the racture duct. This one, it is very important in absorption or transportation of fats. So the fats are being transported independently of the blood. Adaptation of the villus for absorption. So if you look at this, what are some of the adaptations? It's supposed to have some of the adaptations so that it can carry out the process of absorption. Oh, I've not talked about the racture. The epithelium is one cell thick, allowing nutrients to pass through quickly. So that's the meaning. It means that if you look at it here, it's one cell thick. The nutrients can cross, and then they're already inside here. Just one cell thick, you see it. It's just one cell thick. So it allows nutrients to cross very fast. Then number 2, the goblet cells secrete mucus to ensure that absorptive surface is moist, and also nutrients to be dissolved, absorbed. So if you look at it, it has what called the goblet cells. You see the goblet cells. These goblet cells, they produce the mucus, so that now it is at least the surface is moist and the nutrients can be absorbed very fast. And then number 3, it has many mitochondria to supply energy for active absorption of nutrients. So whenever I talk about mitochondria, we are talking about energy. So this helps, or it aids in carrying out active transport. You studied the different ways in which substances cross the membrane, yes, or the cell membrane. You said diffusion, you have active transport, you have osmosis, actually even facilitated diffusion. So in this case, nutrients are supposed to be, some of them, they are supposed to be absorbed by active transport. And active transport is the movement of molecules from region of low concentration to the region of high concentration against a concentration gradient. So since it is from low to high, it's gonna be having a high amount of energy which is needed for it to move from low level of the hill to a high level of the hill. So mitochondria is very important in the production of energy. That's microvilli for further increase of the surface area. So the villus is divided into other small parts so that the surface area for absorption can increase. And then lastly, it has what called the lymph vessel called the lacteal in each villus, which absorbs and transports lipids. You'll find out that I say that this lymph, this lacteal duct, it is very important in absorbing the fats, and then the fats, they follow the lymph duct, and then they go to other parts of the body, or to the left part of the chest. It joins there, and then it joins the heart for further pumping. Number one, we said that it has epithelium, is 1 cell thick. Why is it important? To allow nutrients to move fast or quickly. You talk about the fact, the fact or the part, and then you tell us the function of it. It has goblet cells. How about goblet cells? Secrete mucus. What's the function of the mucus? To moist and dissolves the nutrients, or to allow the nutrients to be dissolved and absorbed quickly also. Then another, it has mitochondria. What's the function of mitochondria? Produce energy. What's the function of the energy? For active absorption. Microvilli, how about microvilli? It increases the surface area for absorption. And then it has the lymph vessel. How about it? This lymph vessel is called lacteal, or it has the lacteal duct, which absorbs lipids and transports it. So transport of amino acids and glucose. Actually, transportation, how does it look like? So you start with the amino acids and glucose absorbed into the blood capillaries of the villi in the small intestines. Okay, once they are absorbed, what happens? The capillaries join together to form a large venue of venules. These are small veins, yes, to form a hepatic portal vein that transports amino acids and glucose to the liver. So it transports this to the what? To the liver. And then glucose and amino acids through the hepatic portal vein to the heart. And then lastly, but nonetheless, the liver converts the excess glucose into glycogen, which is stored. And then the excess amino acids are deaminated. It means that when you eat too many eggs, you don't benefit. You only benefit if the eggs are few. Why? Because the eggs are made up of amino acids, the protein. So what we do is we just deaminate it, and then we excrete it as a waste product. Next time we will start with the